Until now, little has been known about how Borrelia bacteria interacts with cells. In this episode, we are going back to Finland to understand what happens at a cellular level and understand what enables Borrelia to survive. Dr. Leona Gilbert is a docent of cell and molecular biology and the CEO of TESDED. Dr. Gilbert has over 20 years' experience in research-driven initiatives that provide valuable tools for patients. She has a doctorate in biotechnology and extensive experience in bioinnovation and biobusiness. Welcome back to our podcast, Leona.
Great. Hi. Thank you for having me here.
Let's start with understanding the immune system. I know you're looking at cells and various forms of Borrelia. How does the immune system respond when Borrelia enters the body? For example, what does a macrophage do when it encounters Borrelia?
Well, like the macrophage, when it encounters any other pathogen, it will pick it up and recognize it by these patterns of recognition on the surface of Borrelia. And when I say recognize it, it really attaches itself to the Borrelia and want to phagocyte it or want to ingest it, okay, and handle it and resolve it, and thereby actually presenting little proteins or peptides on the surface of the macrophage so that it can communicate to other cells. And dendritic cells do the same thing. They phagocyte these Borrelia because somehow the communication in the immune system to tell the immune cells that they should, be wary that there is a pathogen there, they have to communicate. And the communication starts obviously by processing and resolving that pathogen that they have ingested and displaying parts of it on the outside surface so that it can communicate to other cells. So it's not an exceptional pathway, it's like the normal pathway. But Borrelia is so big. It's around 20 microns in length, but it's very thin, like hair, like it's 20 nanometers widthwise, and it is a corkscrew-like structure. So it is really a large pathogen comparatively to other bacteria like E. coli or even viruses that could be just a couple nanometers, in diameter or 10 nanometers in diameter. So it's a very large pathogen. So the macrophage really has to have unique kind of mechanisms of actually ingesting it and processing it, because it's what its job to do is really to process pathogens. So there have been other groups that indicate that the Borrelia actually in dendritic cells actually drills into the cell. Okay. And other people and us have shown that actually the philopods, so the arms of the macrophages, actually surrounds the Borrelia and forces it to kind of curl up, and then it ingests it into the cell. So there's different mechanisms that these immune cells can actually use to ingest it and then to process it.
Oh, that's really a great explanation. Thank you. And we are going to link to some of the images and the research, obviously, that to your website, so that people that have that aptitude and want to go and see these images can see them for themselves. Now, what is pleomorphism? And again, I know you're looking at these images under a microscope, so I'm really asking you to kind of describe what you're seeing as well.
Sure. So I just described that Borrelia is like a corkscrew-like structure. So it's kind of coiling, and it's really 20 micrometers in length and 200 nanometers width. So it's very thin, okay? And pleomorphic means that it can change its shape based on its environment. So we and others have demonstrated that when Borrelia is faced with an adverse environment, could be a change of pH, change in nutrients, change in media, could be even be the presence of human sera, okay? It recognizes the environment is stressful for them, so they tend to protrude its membrane, outer membrane, and make these little blebs. Okay. And the blebs actually ball out, okay, and expand and make these round bodies. Okay. These round bodies really are circular. They're about 2 microns in diameter. Okay. And inside, you can really see the protoplastic cylinder where the genetic material of the Borrelia is. It's really coiled inside this round body. Other people have said that this is a cell wall deficient form or pleromorphic form, but we have demonstrated that actually the round body maintains its integrity of its cell wall. So it's really not a cell wall deficient. It is really truly a two plasma membrane structure, and it holds its integrity. Besides the bleb, as I mentioned to you, the first signs of adverse conditions or environment, it turns into a round body. But we also have noticed in vitro as well as in vivo studies of ours and others that Borrelia can form biofilm structures. And in an in vitro system, we call them biofilm-like structures, because in a normal culture medium, or like growing these bacteria up over a course of 7 days, they like to clump together. They like to, bring all their community individuals together and form a biofilm. Now, it's been speculated that the biofilms is a really resistant form to protect the bacteria against any, against even antibiotics and so forth. But in that biofilm, it's also, having different pleromorphic forms. So you will see the spirochete form, the parent form. You will see the round body form. Okay. You will see these spirochetes with these blebs. Okay. You will see all of that in the biofilm-like structures. And we really do see this in human studies as well, and in animal studies, and in culture.
Now, you mentioned the biofilms. What are some of the mechanisms of Borrelia fitness that do enable them to survive?
Well, I think the mechanism is that the kind of sense, it's called quorum sensing. So they sense that there is an adverse environment, and they want to clump together to actually provide in a normal, let's say in a normal host, like the human host, they will actually recruit other kinds of proteins there, like to create this extracellular matrix so that the biofilm can attach itself there and start building up really a solid structure. And we have seen these in studies of the joints in people, and also in the brains of individuals of autopsy samples. So it's really creating a really niche for this Borrelia. And through quorum sensing, the whole point is that they can share genetic material amongst each other, okay, to actually allow this kind of persistent form and also survival within the structure. And incidentally, there are other studies that actually speculate that it could also be that these biofilms are a good platform for other microbes, okay, beneficial or not, to actually participate in building up this architecture of the biofilm. So it could be a kind of a synergetic effect that allows kind of its survival with other microbes. But on its own, we know definitely that there is recruitment of extracellular matrix proteins that allows it to establish itself as a biofilm in hosts.
That's extraordinary. Now, yeah. I know you're using a transmission electron microscope to produce graphs. I'm just wondering if you can tell us a little more about that, and what is immunolabeling?
Yeah, so in order to see these things in high resolution, we need to use transmission electron microscopy. So basically, we're sending electrons through samples, like cell samples, okay, and so that we can see the interior or the surface of cells. And it's interesting because we can see actually the architecture of cells or things inside the cells as well. And if we do immunolabeling, which means if we use an antibody that's very specific against Borrelia, or therefore its structures like DNA or the outer surface protein C and A, or other structures like P41 of the flagella, if we use antibodies against those structures of the Borrelia, we can very specifically label those parts, and we can see them in these electron microscopes. And that's what immunolabeling is. And not only for transmission electron microscope, but we can also immunolabel, cells, Borrelia cells or normal cells or macrophages that have ingested Borrelia. We can immunolabel them with antibodies and see the Borrelia inside inside the macrophage or outside the macrophage or any other cell type that we want to investigate. So it's really, immuno means an antibody directed against the very structures of the Borrelia that we want to see, because seeing just microscope samples is very subjective. It doesn't say that you're not identifying that that's a Borrelia. You have to use some other technique, either label its proteins or label even its DNA with probes to see that that's the Borrelia definitely inside a certain cell type. So that's why we use these immunolabel techniques.
I found it interesting that you're examining survival of Borrelia cells. What did you find?
Yeah, it's really crazy. So when we first wanted to understand what happens with Borrelia in our immune system, we first investigated macrophages. And we wanted to see the route in which macrophages can resolve Borrelia. And that was us and others have published that really macrophages are good phagocytes, which means that they ingest the pathogen and can process through this endocytotic pathway that involves lysosomes that have really a acidic pH. Okay. But when we were thinking about how does Borrelia actually establish itself in the host, in the human, we decided to use two cell lines, so the skin fibroblast cell line and also the chondrocytes. So chondrocytes meaning joint cells, okay? Because we know that Borrelia obviously infects the skin and needs to go to it can go to the lymphatic system and also can go through our blood system and disseminate to other organs. We know this, but we also know that Borrelia can hide out in niches like the knee, so synovial sites. And that's what prompted us to investigate. So can these cells resolve the Borrelia, or can Borrelia survive within the cells? And what we have done is we have infected both the skin cell, okay, so the epithelial fibroblast cells, and we have infected the synovial sites, so the joint cells, with Borrelia at different time points from 24 hours all the way to 9 days. Okay. And we observed kind of what the pleromorphic form of Borrelia was in these cell lines. Like, do these cell lines, because it's a harsh environment or a different cell line that they're used to, will it create these pleromorphic forms as a mechanism to adapt to that type of environment? Or what really happens to them? Are they resolved by these cells? And it's interesting that what we have seen is once we infect the cells and we wash off the cells after a certain time point, like 24 hours, and if we leave the infection to go on for another 48 hours, let's say, and we break up the mammalian cell, so the human cell, and we let the Borrelia culture to grow, we can culture Borrelia from the inside of these cells. And this is this is really an indication that the Borrelia can survive in these cells. And I'll bet, okay, the two different cell lines have different time points in which the Borrelia, it takes for the Borrelia to kind of get accustomed to its environment so that it could actually be cultured out of it. But still, the Borrelia can survive in both of these cell lines. And that's really interesting because, well, we know that these cell lines are not professional phagocytes, which like the macrophages. So it's not used to digesting pathogens and obviously resolving it. But you would expect, we thought, that these cells would be infected Borrelia kind of, how do I say, kills the cell, okay, because it has to replicate. It needs nutrients to replicate, and like a typical other bacteria like E. coli or Staphylococcus pyogenes or any viral infection as well. They take over the cellular machinery for its replication, and they usually tend to kill the cell. And when they kill the cell, then of course they can spread the infection. But Borrelia is not killing the cell. So it's really interesting that it hides out in these cells, okay, and maybe to protect itself, it does form different pleromorphic forms, okay. So we see more round bodies or coiled up forms in synovial sites comparatively to the skin cell, And that's really interesting because the synovial environment is more acidic, okay, and it's definitely different than the skin cells. So it's really interesting that the Borrelia is really surviving
Truly,
within.
that's extraordinary.
Yeah, it's crazy. This is really an amazing bacteria.
Yeah. And are other scientists replicating that research as well?
Yeah, and like it's not just us. Others have demonstrated that in different types of cell lines, like the fibrobros, like this absolutely supporting other people's research. The synovial sites was something different, and we had to really justify why we were using the cell line. But I'm sure other people are looking into replicating this work. And besides, we have keep on replicating it to make sure that the methods that we're using are demonstrating the same results that we are getting.
Yeah,
So,
exactly.
Yeah.
Now, in one of your experiments, you were able to stain for cell death markers for apoptosis, necrosis, and dead cells. Can you explain that experiment and the results?
Yeah. So I think we're talking a lot about the macrophages. So there's different types of cell death, and simply put, the organized cell death is apoptosis. Okay, which means that the cell is undergoing programmed cell death, so that it's not creating a whole pile of garbage that's thrown into the environment of where the cell is dying from. Okay, so it's controlled, meaning that when the cell dies, it will form up these vacuoles. Okay, and they are surrounded by actually a double membrane, and annexin 5 is a marker that indicates that actually this is really apoptosis. Okay, the other pathway of cell death, and there are different pathways, but the ones that we were interested in is necrosis. Okay, and that's like tissue damage. Okay, and tissue damage is not controlled, it's not programmed. It's when accidentally you damage the cell, or in some cases the viruses will replicate so fast that the cell will just kind of burst open. Okay, and all that garbage is of the cell, the inside of the cell is kind of spewed into the environment. Okay, so it's uncontrolled death, but apoptosis is controlled death. And in the macrophages, one of the markers, the annexin 5, compared to cell death markers, which obviously there are DNA stains to show this, there's also vacuole markers to show this as well. So we can really differentiate which cells are going under necrosis, a pathway, in which cells are going under apoptosis through annexin 5 staining. Yeah. So in macrophages, it's controlled death in some death, okay, but it's a lot more. There's more death in monocytes than professional macrophages. So, and that kind of makes sense because monocytes are naive cells, which means that they're not mature. We call them the teenagers of the macrophage. Okay, they don't have their final, function yet, but macrophage has its final function, and that's to resolve the bacteria and to display peptides or proteins on its outside surface, so it can start communicating to, let's say, T cells, B cells, those kind of things, and mostly T cells. Yeah. And so the point is that these annexin apoptosis pathway labeled is what we see predominantly in macrophages. We tried to use the same kind of technique for the fibrobose cell line and the sarcoma cell lines, or the joint cell lines, but obviously they weren't causing cell death. So we had to kind of think, okay, they're surviving. So they're not undergoing apoptosis or necrosis, they're surviving. So obviously we had to change our thinking and discover that, okay, what is really happening in these chondrocyte cell lines and the fibrobose cell lines. So yeah. Wow. That was interesting because our hypothesis was wrong. We thought that apoptosis would have happened in these two cell lines, but that was not the case, actually.
Well, that's an important discovery too.
Yeah, it was. It really was.
Now, what are persistent tissue reservoirs? And is it possible that Borrelia resides in some of those places? I mean, some that, I don't know, came to mind. Obviously, I saw you speak before about vagus nerve and I'm just curious about lymphatic and glymphatic tissue as well.
Yeah. So these persistent reserves, these are these niches, okay, of where Borrelia wants to hide out. Okay, and there are other people really publishing on great science that actually indicates that Borrelia has been seen in lymphatics, in the lymph nodes even. Okay, so it's really, and it actually kind of downregulates the maturation of B cells into plasma cells. So they really can affect the overall kind of antibody production pathway that's occurring in the host. And that was a really interesting finding. We also know the niches like the knee, the eyes, the brain. You mentioned the vagus nerve. I don't study the vagus nerve, but I have read the papers that have indicated that this could be a niche as well. And it's really trying to get the Borrelia to, or the Borrelia is trying to, evade the immune system. So it needs to hide out somewhere, because there is that question, as you know, of, what is the mechanism behind post-treatment Lyme disease syndrome or chronic Lyme, right? And yeah, and this leads to the whole thing. Is there a persistent infection, or is there persistent antigens, or is it an autoimmune current, or is it an immune dysfunction? So there are evidence to suggest that these persistent kind of niches do exist in not only animal studies, non-human primate studies, and also human studies. So there is these niches there, and we just need more research to really show that the persistence is happening. So yeah, this is a leading hypothesis of why we're seeing a lot of patients suffering from post-treatment Lyme disease and chronic Lyme.
Now, is there anything new that you've learned about Borrelia and the immune system?
Yes. It's so interesting that, one of the emerging areas of research for a lot of pathogens is these extracellular vesicles. Extracellular vesicles are these vesicles that the pathogen will pump out, okay, that have components of itself inside it, okay, and they can act as a decoy to the immune system, or they can be responsible for even having, even establishing kind of the first immune reaction, okay, in some hosts. And we have demonstrated, us and others have demonstrated, that actually Borrelia can produce these extracellular vesicles, okay, and inside these vesicles have a lot of, how do I say, antigens. And antigens are those proteins, okay, from the Borrelia, or entities from the Borrelia that actually the immune system recognizes as foreign and can elicit an immune response. Okay, so EVs are now being linked to autoimmune diseases, to cognitive diseases. Other EVs are extracellular vesicles are being linked to how these pathogens can evade the immune system. So it could be a decoy for the Borrelia to evade the immune system, meaning that once it gets into the blood system or into the lymphatics, it can push out these extracellular vesicles and trick the macrophage, okay, or the dendritic cells, that, hey, these EVs are the Borrelia, not, the full form of the Borrelia. So it can be really that these are acting as a decoy so that the Borrelia can go to the sites of invasion, like joints and so forth, and disseminate. So we have demonstrated that the certain proteins like the peptidoglycan, glycoproteins, the P39, OSPC and A, as well as DNA. Okay, so DNA, Borrelia DNA is in these outer membrane vesicles, or these extracellular vesicles, and and yeah, so we are undergoing studies to demonstrate that they could, behave as a decoy for the immune system.
That's interesting. Now, what have you been learning about phytochemicals?
Yeah. So I like, by default, I'm a scientist, so I need to try to, learn different areas about, tick-borne diseases. And obviously, because of patients suffering so much, I wanted to understand what they're going through, and is there any treatment, like, why is treatment failing, and are there other treatments that they are, provided? And one of the things is phytochemicals or herbal supplements. Now, Now, I think it was in 2018, I presented phytochemicals to the German Borreliosis Society. And then I realized actually there's a lot of good anecdotal evidence, okay, through
Mm-hmm.
books or through gray papers or through blogs that demonstrate that there has been success in the use of phytochemicals in Lyme patients. But when you actually look at the peer-reviewed publications, okay, and the actual types of experiments that they're using, and to evaluate actually, is there really an evidence-based approach to the utility of phytochemicals or herbal supplements and Lyme disease. And I was really quite surprised and disappointed because there's a lot of in vitro studies, so test tube studies, okay, that you put the phytochemical onto the Borrelia and you look at it to see if it kills it or doesn't kill it or the strength of the killing and so forth. But there is no clinical study, no human clinical study.
Right. Yeah.
And I'm thinking, we're doing such a disservice. This is my opinion. We're doing a disservice in not providing this evidence, because I know some clinicians have to justify the treatment protocols to their health authorities, their national health authorities. It would be so simple as a defense, and also supporting, okay, in their protocols, if they had an evidence-based article that actually demonstrates the effectiveness of these phytochemicals. So this is an area that we need absolutely to do more research. And I have heard and listened to people's comments about why there isn't, or the speculation of why there isn't these clinical studies, and they say, well, it's too time consuming, it's too expensive, but actually it's not. It's something that could be easily done, because they're using it on their patients anyway. So we get ethical approval.
Right.
We have controls. You treat your patients, you follow them over a course of a year and a half. I think it should be a year and a half or longer, not 12 months, but a year and a half or longer. And you look at their outcomes. You can look at their outcomes quite easily through validated surveys on their symptoms, or also their blood chemistries, or also their test results. So it is a quite straightforward kind of, in my mind, experiment to do, okay, study to do, and I'm encouraging a lot of clinicians to do this, and I have convinced some clinicians to do this. So hopefully in the next year or two, we'll start seeing some of the data being published on the use of phytochemicals and their patients. So.
Great. Thank you. Now, I believe you or someone on your team might be attending the upcoming crypto infections conference in Dublin in June 2023. What are crypto infections? And can I get rid of them if I buy Bitcoin?
Yeah, I know. It's such a great name. Yeah, I think it was more the politically correct word to demonstrate chronic infections. Yeah. So I think I think it was coined by Christian Perron. Okay. And I think it's a one way of popularizing this area, because it's not just Borrelia that causes chronic conditions, but it's also other pathogens. So I think that was the point. Yeah.
Yeah, well, I mean, we'll be watching that, because I'm sure there'll be some very interesting presentations that come out of that.
Yeah, they have a really good speaker group. So I will be there, absolutely, cheerleading everybody. We have some posters from medical students coming out giving posters, and some from other scientists and clinicians. So it'll be interesting to see what the emerging research in this area is as well. Yeah.
Great. I hope there's some wonderful synergy there that just leads to many, many more discoveries and solutions.
Yes, me too. Me too.
What can you tell us about Tested?
Tested. Yeah.
And what is unique about your testing kit?
Yeah. Okay. So Tested is a university spin-off company from Uvasco University. And we decided to start a company because we realized through European Commission sponsored projects that the testing for Lyme disease is really, really poor, meaning the sensitivity and specificity is poor. And they're only testing one microbe at a time and one antibody at a time, which indicates one disease stage at a time. Right. So really the whole world is using the two-tier system, basically. And we think that this is failing the patients, because within my PhD students' PhD thesis is we demonstrated that actually these patients are not suffering only from one microbe like Borrelia. They're suffering from other microbes like Bartonella, like Babesia, and it's interesting that especially in chronic patients, they're suffering for a lot more pathogens than just somebody being bitten by a tick and getting Borrelia. And that's the whole point, that we surveyed, I don't know, 15,000 patients till now, and against 20 different microbes, and we statistically wanted to provide a test kit that actually was really efficient for these patients. So our test kit is unique in the sense it's called TickPlex. It tests for 15 different microbes. Okay, the most statistically significant microbes that we demonstrated, and it tests for IgG and IgM at the same time, separately, of course, but at the same time. And we have quantification of this immune reaction, so you can see how strong your immune reaction is based on the antibody concentration. And we have correlated that to different disease stages, like acute dissemination, late dissemination, and then chronic stage. And we have this saying that Borrelia likes its friends. So Borrelia and friends, yeah. So we demonstrated that 85% of Lyme patients in that study, and even now in other studies, actually have co-infections. We have shown this in Canadian patients, European patients, American patients, and Australian patients as well. So look for our Australian paper coming out soon, because yeah.
And does it also include testing for mycotoxins?
Yeah, so microtoxins is a new product of ours. It's called ToxyPlex, toxins, and it really is the toxins for mold. So we're detecting directly these toxins, mycotoxins, in the sera of the patients. Again, another area of research and another area of concern is, are these patients suffering from other things besides infectious material, like heavy metals or mold toxins? Right. Yeah. So we have developed, in a collaboration with San Aviv Medical Institute from Mexico, this ToxyPlex. Yeah.
I know them.
Yeah, they're really great. It was a really great collaboration, and we're testing the most 5 known and pronounced mycotoxins that are found in food and feed, because how do we get mycotoxins? We eat them, okay, through our food and feed, and we also obviously can breathe them in, like the sick building syndrome, so those buildings that have mold inside them, and so the patients are suffering and they're inhaling these mycotoxins, these spores from the mold. So we really, it's one of its kind. We are testing directly 5 different mycotoxins in the blood of patients, and yeah, in a very simple ELISA format, or a very basic format, so you can get your test results within 4 hours instead of 2 weeks. So
oh wow,
it's really great. We just launched it last fall, and we have Armin's lab using it, and we have, of course, San Aviv using it, and of course we're trying to spread it to our other customers as well.
And then, for other customers, how can people around the world access your various testing kits?
Yeah, so while we wait, okay, until we can get into Canada, and we're saying while we wait. So we have a new laboratory opening up in the States. So I understand that it could be easy to move samples from Canada to the States, and they will be opening up at the end of June. So watch my website on that, because you will see.
Great.
But in the meantime, Armin's lab is accepting, because they are now CLIA and CAP approved. Okay, this is the designation, the highest designation in the United States. Yeah, so they can receive samples from Canada and the United States, and they have a really good transport system. So you can go to Armin's Lab website, and you can just order the test kit, ToxyPlex or TickPlex, from them, and they will send you a sampling kit, like a blood draw kit, okay, with its tubes, give you instructions on how to send it back, and they will do the testing quite robustly. So
Great. Thank you. Now, we are getting close to the end of the interview, but I do want to ask you, because I know you've worked a lot with patient groups when you're conducting research, and I just thought I would get you to comment about, working with patient groups, because obviously we want to be involved in the research and we want to participate and we want to be part of the solution, most importantly.
Great. So, yes. I have to say that we cannot do this without the patients. I mean, I can't do anything without the patients. So it's very important that we have good kind of involvement with the patients groups. And I work closely with a lot of patient groups, as you mentioned, but the one that I want to mention right now, because it's called Global Lyme. Okay. And Global Lyme is kind of like-minded individuals from patient groups come in and we discuss different things. Okay. And the participation of this Lyme Global is only up to the amount of time that you can put in. But basically we have minutes. Okay. So if you can attend meetings, you can read the minutes, but we go over such research projects, okay, to get patients involved. For instance, by the way, I'm putting together a consortium to apply for European Commission funding, and I've just talked to Dr. Amir Kadir in Quebec, and obviously if Canada is involved, we need Canadian patients. Obviously. Right. So we will come back to you guys, okay? But I really encourage all patient groups and and members that if they want to know what's emerging, the research and what other patient groups issues are, you have the same issues. It's a global issues. It's not like we're not exceptional in the fact that governments are or clinicians are turning away, the patients, or that chronic Lyme isn't being, is not believed in. There's different terminologies for chronic Lyme in different countries. And the point is there's same legal, constitutional issues in a lot of these countries. And Lyme Global is the one that helps us, air out these issues and maybe learn from each other as well. So it's not just patient groups and patients that are involved. There's like me, like-minded individuals. Professor Jack Lambert is there from Dublin, from LRC, and Can Lyme, you guys are there too, but we need to see your faces.
Now, one final question.
Okay.
Is there any other emerging research you're excited about?
There's always. Yes. So,
and I know you've got some publications coming up, so we won't spoil anything.
No, but I can just say this. I have encouraged a lot of clinicians to do to bring their cases into a format that we can actually mine it. Okay. Bring their patients' data into a format that we can mine it. So you will be seeing a lot of publications coming out that's using machine learning and AI to really statistically cluster and support our hypothesis that chronic Lyme does exist, and these are the characteristics, or there's immune dysfunction, and these are the reasons why, and so forth. So AI and artificial intelligence and machine learning you're going to be seeing more and more of those published. Yeah.
Well, thank you so much for your time. I know you've had a very long day over in Finland. I appreciate you staying and keeping fresh so that you could do an interview with us. And we will certainly stay in touch with all the research you're involved in.
Great. And my pleasure. Thank you for inviting me.
My brain is full. It was so fascinating to learn about Borrelia and how they survive. Let's hope that this leads to further discoveries in how to change the environment and eradicate them from our bodies. That's our science fix for today. Stay safe in the outdoors.